but we've just kind of finished doing some other work about network comparison, so I'm going to talk about that at the end, basically because I think it's exciting because I've only just done it. So really, first of all, what I want to talk about is what we want. So in most of the world I look at, I'm really interested in biological data, so I'm interested in protein interaction networks, but other types of biological networks. And what you have is this big, massive network, and I'd like to extract pieces out of it that mean something. And that's quite a big sentence, because how do I know when I've taken a piece out of it that it means something? Because it's very easy. There's lots of mathematical techniques for decomposing a network, and I'll touch on some of those. But what I'd like to know is that the things I've decomposed it into mean something biological. So that's the problem, the basic problem I'm going to try and answer. So just in case anyone doesn't work on these or doesn't care about them, I'm just going to make sure that everyone's in the same place I am as we go through this. So protein interaction networks. Proteins are the large organic molecules in your cells, in every biological cell that does stuff. So they are the actors in this, and they carry out duties as encoded by the genes. And a protein interaction network, the nodes in here, so the blue dots and the yellow dots, are proteins, and the interactions are done as edges between them. And there's lots and lots of experimental data on this. So you can have a yeast interaction network with 6,000 nodes in it, so that's about the number of proteins in yeast, and 30, 40, 50,000 edges, that kind of thing, and that's all experimental data. They're usually undirected, but the edges may or may not have weights, so you could put a probability on them of how likely that edge is to exist. Everything I show you today you could do with weighted networks. I'm not going to do it with weighted networks because it makes it ugly, but basically the principle remains the same. So if I'm talking to biologists, what they tell me they want is, from that mass of data, is something they would call a module, so a section of proteins, a set of proteins that go together to perform a particular function. So what I want to be able to do is identify novel modules within the protein interaction network. I don't want to identify a module they already knew existed. That's actually relatively straightforward, and there's lots of people who've published very good papers about doing exactly what I've just said. But what I want to do is try and find modules that do contain biology, so they're real in terms of they have a coherent function, but nobody knew that. You could only find that by doing a mathematical deconvolution of the network. You couldn't find it just by, I know what the labels already are on this network, and these 5 will perform this function, therefore module. Yep. So we're trying to look at it in a slightly different way. Now, the default way, if I start describing that problem, I'm talking to a mathematician or a statistician, is they go, well, what you want is a community detection method. So you want a method that basically says that the things within this community are more connected to each other than they are to anything else. It's a very standard technique, and there's lots of ways of doing that. One of the things I should state here is that in biological terms, we don't know if that's actually what we want. So mathematically, there are solutions or different ways of solving that. Actually, I'm going to use community detection methods. But strictly speaking, I don't need to for what I'm about to do. But we should also be aware that if you're thinking biologically, it's not totally clear that when I want a module, I definitively want something that's connected up in that way. It just happens to be that's the mathematical way we've constructed breaking down networks. What I really want when I want a module is a set of things that share a common functional annotation, so that they all perform a function, which is different, hopefully, or can be separated out from the rest of the network. So if I just took that set of proteins, that set of actors, they do something, and everything else doesn't do it.
They do something when taken as a collection?
Yes, as a collection.
As opposed to individual?
Yeah.
Yep.
And the kind of way most people do this, if you just ask this question, is they look for something either like functional enrichment, and then or sort of functional homogeneity. So you say the functions of everything I've got in this grouping are very similar, so that makes a sensible grouping, and that there's an enrichment for that function in this grouping versus the functions I have spread out over the entire network. So if you ask this in a kind of different way, if you said, did all these people go to the same university, and that was spread across a network, if you were trying to deconvolute it, you'd want to pull out a group that all went to UCL, with very few people who went to any other university within that group. And that's the same kind of idea that people are using when they're doing this for biology.
I don't want to bother you too much, but that example is each of the individual nodes has the property all by themselves, though.
Yes, so this is where this is a bit difficult. So functional annotation in biology tends to be annotated on the nodes, but we know that most processes are not carried out by a single node.
Okay, but the annotation is node specific.
Yes.
Thank you.
It's one of those where that's how they've annotated it, but the actual...
That's their job, right?
Yeah. So the final part of this, which I think is something that kind of became obvious maybe 5 or 6 years ago, but up till this point people actually hadn't even really thought about it, was that it isn't clear that there's a single decomposition of the network that makes sense at all. Now that's very easy if I talk about social data. If I say you have a family, the family would all live in a village, which would probably all be inside a county, which would all be inside a nation. Yeah? And so it depended whether I wanted to explore nations or families as to how I would split up a network. In biology, it's worse than that. So I can give you an example here. So this is actually part of a biological network, and I've taken out all the nodes. Basically, it is a community, but it relates to a whole set of nodes which all have something to do with the ribosome. But then I could split it another level, and the yellow ones are mostly the ribosomal subunits, the reds are the precursors and the processors, the small subunit, and the blue are the large subunit. So there's a scale there that you can see. So if I wanted to find out about the ribosome, I'd want a different kind of scaling of how I cut down the network from if I wanted to find out the processes inside it. The problem is, if I say I don't know what biological process I'm looking for at the beginning, then I can't tell you what scale I want to look at. I don't have that information. So everything I'm going to do, I'm going to have to do across all scales, because otherwise I might miss something very important. Because actually we don't know the level that biology is working at, so what scale would be appropriate. We know for specific cases that we have understanding of, but not more generally. And then I think I've kind of mentioned this already because I've been answering questions and stuff as well, but this is the idea. So if I have a network, and if I'm sort of increasing the resolution, so I'm zooming down on it, what I'm hoping to find are groupings like that grouping where I've got those 5 squares all grouped together to be able to say that they have a functional similarity and therefore they perform a function that's correct. If I break up the network too much, then I won't see anything, and if I leave the network too close together, I won't either. But actually, the biology could be at different scales, because it could have been that I could have found that first community at a much easier resolution, and then I don't find the others until a slightly higher resolution. So it's not clear when these things should happen.
Is there any theory, as in from the biology, any theory of what scale things should be operating at?
It depends on which biology people are interested in. So actually the answer you usually get to that, which I quite like, is between 6 and 35 proteins. And then when you ask them why that's the answer, because that's what they can deal with experimentally. So that is probably, I mean, I think it's a reasonable answer, because it's then, if you give me a community that has between 6 and 35, I can do something with it. But there are definitely some functions. I mean, that ribosome thing I showed you just then clearly had far more components than 35. But they kind of, that's the sort of answer you get. So the last thing I should mention is, I'm back at that snapshot of the pin I showed you before, and it's kind of an obvious comment because I'm dealing with biological data, but it's true on most networks. I actually don't have annotations for all the nodes, because if I did, I kind of wouldn't need to do this, because then I could just work out where the communities were because I knew what all their functions were. So I have nodes without annotation on, so in this case it's those black dots. And one of the ways I want you to think about the problem I'm doing now is, imagine that I had all of the yellow dots were labelled exactly as they are now, so there's some black there, and the blue ones were labelled, but none of the red ones were. So the question is, could I know that the red ones should form a community, but there's basically no annotation in there? Because actually it's more likely, if we look at the way people currently do it, that what they do is they look at a slightly different resolution, and the red ones would half end up in the yellow community and half in the blue one. Because it's a much more satisfying answer than, here's a load of stuff, I don't know what it does. Yeah? And so people tend not to head for that answer in all of these problems. So then I'm afraid I have to do a little bit more biology just to explain how the problem is set up. So the annotation on the nodes we use is something called the gene ontology. This is very standard for annotating protein interaction networks. And it's a set of concepts, and the relationships to each other are arranged as a hierarchy. So this is just a tiny part of the biological process, gene ontology. So if something's labelled, say, right down at the bottom here with negative regulation of signal transduction, it will also have all the labels back up the tree all the way up to having a biological process. Yeah, it's the way the labelling is done. And there are different types of connection in here, so you can have things like is part of or is not part of, and all these kinds of things. But basically what really happens in terms of a protein interaction network is that proteins are not only necessarily not labelled, but they're labelled to different depths within this hierarchy. Because of course, some proteins we've done lots of experiments on, so I'm able to tell you that it's a negative regulation of signal transduction. Much more commonly, I'll be able to tell you it has something to do with signal transduction, because some experiments have been carried out. And then, I might be able to tell you it only has something to do with signal transmission, or maybe I just know it has something to do with signalling. And of course that's a problem, because then instead of having these nice clean, it's a red dot or it's a yellow dot, it's a kind of red-yellow coloured dot right from the top, or it's a bright pink dot, because it definitely is one of these. So that's another factor you have to take into account. And actually, it's very clear on biological networks, but all the other types of network data I've worked with, it's very similar. If you think about the labelling, it's never this very tidy, everything is labelled to exactly the same depth with exactly the same amount of knowledge. If you take a social network, you're usually taking the labelling that people have placed on it themselves, which can often be to very different levels of information about what they have or have not told you about.
Quick question, it doesn't contain your nice idea of scale, but if you look at the mixed membership models of Aroldi, etc., where with a certain probability for a relationship, you're in one or other of the groups that exist, how would that relate to what you're doing?
So that's kind of the, if things were labelled, then you could build your communities effectively based on the labelling. What I'm really interested in seeing is, can I design something such that it does that correctly without using the labelling information in the first place, and then the other areas that it picks up where there are no labels will also be a biological process. So if somebody solves the function of one protein within that module, they would know what the process was for all of those in some sense.
I was being confusing, sorry. I more asked, like, not looking at the fact that some of your observations didn't have labels or did not. If everything was unlabeled and you know there are communities behind it, what if for a certain relationship you be belonging to one community with a certain probability and you mix over the community? So maybe I'm always in the statistical science department community, but maybe you're half the time you're in a biology community and half in another, right? Then your relationships, the group you behave like, there will be a probability you'd be like one or the other. How would a model like that, which is the mixed membership of Aroldi, relate to what you're trying to do if we take out the fact that you sometimes have labeled or unlabeled data? Let's remove that.
Okay, remove that. I guess it would be in my head, it would be a slightly different way of representing how this is represented, because you can see within this that you can have a process down here that belongs to multiple.
So it's the hierarchical aspect.
It would be the hierarchical aspect that would relate.
Okay, sorry.
That'd be the way I'd think about it, I think. So, as I just said, and it's probably one of the most important problems in this, because it's the one that if you read the literature has not been dealt with, there's a huge bias in protein interaction networks, and it's general within biology, because in general labs in biology, and this is not to complain about them at all, because I would not want to have to do their work, it's much harder than mine, but they work on a particular protein or a family of proteins or a particular function, because obviously it's very hard to work on everything all the time. And what that means is that there are areas in the network or proteins where we know a huge amount. So there's a huge inspection bias in this. And that means it's actually quite easy to do what I showed you at the beginning, find communities like this, because lots of people have worked a lot on the ribosome. So they found pretty much every protein that has something to do with it and labelled it beautifully. But there are lots of other areas where people haven't worked on them so much, so they're not labelled at all. So one of the things we have to do is, if we're calculating how similar two proteins are, we have to use an information theoretic technique to get rid of that bias. I'm just very quickly going to describe these. So you can just look at something like the Jacquard index between the sets of terms. It turns out that that's kind of okay, but doesn't really deal with the problems of the structural ontology and the bias of it. Turns out the best of these is something like the Panday measure at the bottom here. So a protein tends not to have just one function associated with it, it has a set of terms associated with it. So you compare the information content of these sets of terms and look at how that compares to what you would expect if you picked two sets at random from the set you've got. It's quite nicely set up. So I now have measures of how similar two proteins are. Those are quite standard. I have a network of interactions and I have them annotated. So generally what people have done then is run a community detection method and calculate the functional homogeneity of the communities that they pull out. And then they go, the ones that are interesting are those whose functional homogeneity is greater than the median for the network. So this is exciting because these 20 proteins are better than a random set of 20 interacting proteins. Makes kind of sense. So does that work? Well, it sort of half works. So I've said that there's this kind of inspection bias that particular proteins will be investigated more. It's actually worse than that. What there really is that there's an inspection bias that relates to the network structure itself. So it isn't random across the network what's been inspected and what has not. It is very concentrated in certain areas. So this picture here is just a small part of a protein interaction network, and the green proteins are things that have really good labelling, and the purple proteins are things which have very poor labelling. Now, fairly obviously, if that's the kind of pattern I have across the network, what happens is this, that I'm far more likely to have functionally similar proteins in places where I have lots of annotation. So if I then compare that to the median of the network, the only places I'm likely to find communities are where I knew they would be in the first place. It's kind of impossible to find communities. And this is a fairly obvious statement, but it turned out that if you look in the literature of people doing this, that's exactly what we've been doing, repeatedly. And of course, then we say we're doing very well, because everything that we find is very well annotated, and we can say it was a very good thing to find. So it looks great. The problem is that it doesn't tell me anything about, say, this purple area down the bottom. I'm just like, don't know anything. So it kind of defeats the object of attempting to do this in the first place. To put some numbers on what I've just shown you, so this is a particular protein interaction network, the HINT-P network, and these are the different functional similarity measures. And these are the kind of correlations between the functional homogeneity and the fraction of annotated proteins in that random walk. And it's very obvious that this is what you get. So you get this very strong correlation between these things. You get no negative correlation with functional enrichment because it uses a p-value as its measure. It's also noticeable that it's, if you like, strongest with the Panday, and this is partly because the Panday is a much better measure of whether things are functionally similar. So the distributions of SIM-UI and SIM-GIC are really quite weird. I won't bother showing them to you, but they have this very odd distribution of very high scores and then weird bumps in the middle, and so it's very difficult to say exactly what they're doing.
What are those things? Panday, SIM?
So those were the 3 different information content measures of just functional similarity. So they're just different ways of doing it. Doesn't really matter. And this is just 2 different lengths of random walk. What I find slightly disturbing is as the walk gets longer, this is just 3 and 6, and we have done it for a bit longer, the correlation becomes stronger. So actually we're mostly interested in things which are maybe 10, 12, 15, 20 proteins, and at that point you're really only going to find what you already knew. You don't stand a chance.
Is there a theorem about that? And for some random, it seems like there'd be a theorem for some model of random graphs.
I don't know, actually.
I bet there would.
I think you're probably right.
Could do such a thing.
I think you probably could.
Okay.
I don't know if one exists.
Thank
I
you.
actually just don't know.
Like the counts themselves converge fast. So if you have an Erdős-Renyi graph, the counts themselves converge faster if the motif is longer, which is very counterintuitive to me, but it's all of us 1982 or something.
So what I want to be able to do now is evaluate communities which I couldn't evaluate before because I just didn't see them, they didn't exist. So what I'm going to do as an input is any set of partitions of the network. I don't actually care how you broke it up. I'm actually going to use 3 or 4 different community detection measures, but whatever you like. You know, if you want to do little kind of ego networks, one-hop networks, fine, doesn't matter to me, your choice. And you can have any measure of homogeneity of the community. I'm going to use these because I'm working on protein interaction networks, but it doesn't matter. But that's what I need. I need partitions of the network, and I need measures of the be able to measure that homogeneity. And then what I want to output is a probability that that community is a functional module. And I'm going to go back to those random walks to do this. So if I wanted to evaluate that triangle, say, within this network, and it has a functional homogeneity value of 4, I always think this is a very simple process, but it's quite satisfying the way it works. What I'm going to do is I'm going to perform short random walks from each node in the community, which are length 3, because the community is size 3. And a random walk is terminated when it's visited that number of distinct nodes. It can, of course, visit the community itself. That would be an allowed random walk. I'm not going to stop it from doing that. And then you can think of each random walk as an alternative community in that neighbourhood.
Mm-hmm.
That's basically all it is. So it's an alternative choice of community in that local network environment. So if I'm thinking about this now, so I've done my random walks. So I've got these different random walks around. So I now want to calculate how good my community is. I'm going to calculate something which I'm going to call a t-value, because it's basically the tail of the distribution. So I've got the number of random walks with a functional homogeneity greater than the community. Yeah? And the total number of random walks I did. So in this case, my t-value would be 0.5, because I found one random walk which had a greater value. So that's the functional homogeneity of 5, so that's 1 plus 1 on the top, and I did 3 random walks in total. So it's a very sort of straightforward measure if you think about it in those terms.
Mm-hmm.
So if I show you it pictorially, if you start doing lots of random walks, this is what the kinds of things you get. So on the left-hand side, what's happened is you've got a very low t-value, because actually hardly anything that I did a random walk around had a better functional homogeneity than the community I was interested in. Or you can have a t-value where actually a community pretty much looks like every random walk I did around it. Yeah? And as always, remember that the community itself can be one of the random walks, so you can see that kind of spike right next to that value suggests potentially actually we walked the community a few times here. So the problem now is that this is fine, but how many of these random walks do I have to do to have any idea whether this t-value means anything? Because if I just do one random walk and it's worse, that's clearly not enough. But do I need to do 1,000,000 to have any idea, or is it actually possible to get a bound on this so I know that I only have to do a certain number of random walks?
So you started with the triangle.
Yep.
And you did a bunch of random walks.
You start from each node in the triangle maybe lots of times and do random walks of length 3. Yeah.
And that's that histogram then?
Yes. Yep. So it's, I mean, like I say, it's kind of quite pleasing because it's quite straightforward.
And so if the FH values were independent, we'd know the answer to your question.
Yeah, but they're not.
Unlikely that they are. So all we need to do is know that dependence. Okay?
Yeah, if we could do that
Right.
in some way. So in order to try and work this out, we're just actually going to see how many random walks are needed just to stabilise the t-value. So can I stabilise it? So the number of nodes is visited in the random walks, so you have the number of nodes in the community, and then the number of random walks per node in the community. So what I'm going to try and do is calculate a value for z that gives you a t-value stability, and I want it to be independent of community size. And this is kind of useful to me because the biologists don't know what size of community they want. So there's probably a good way of doing it with a dependence on community size as well, but something we wanted to avoid to make it as easy as possible to be able to estimate this for them. So you can just look, which is always a good place to start. So what we did was we did lots and lots of random walks around sets of communities to kind of see what happened with the t-values. So this is just some examples here. And it turns out that once you've done around about 100,000, so if you've walked through 100,000 nodes, yeah, your t-value standard error is about 0.005. So probably that's good enough to be able to make an estimate that, so if two things are that close in t-value, then you wouldn't be able to tell the difference between them, but otherwise we're probably okay. And we're really not going to be able to say that something is significant from this because there's no significance test in this. We're effectively going to be able to rank things in some way. So things with better t-values we will rank closer to the top. But that's as far as we can get at the moment. I'm just going to mention one other thing because I think it's important, that of course random walks can fail. And because I actually want people to use this, one of the things we had to write into this was that if the random walks keep failing, just stop. It turned out this is, because it doesn't happen very often, but when it does, it can make people quite angry because it runs for a very long time at that point. So just to kind of give you an idea of what happens out the other end. So these are two different biological networks. They're both human protein interaction networks, the HINT-P network, which I've kind of shown you before, and biogrid AP. So they've got between 10 and 15,000 nodes. And the biogrid AP has 165,000 edges. And those are their densities and the average degree. And I'm going to assign each protein either to a single community or potentially to multiple communities. There are plenty of methods that do overlapping community detection. So I've got 4 different methods of splitting up the network there. They can all be done at multiple resolutions and scales because there's no known resolution for biological relevance. And I've going to use 3 different similarity measures because, as I said, theoretically I don't care about any of those things. So it's interesting to see whether that's true with everything I've said. So what am I going to compare? Well, I'm going to do a very simple comparison because it's an easy way to look at the data. If you're actually using this to investigate biological properties, you'd probably pick a lower t-value than the one I'm picking because you'd go further up the scale. But a kind of qualitatively similar way to think about this is take CommWalker communities with a t-value less than 0.5, so it's better than half the things around itself, and communities whose functional homogeneity is greater than the median semantic similarity of interacting proteins, because that should be about half the things in the network. Yep. Now, obviously, it's not exactly the same, but qualitatively it's the same. And I can think of there being 4 sets of communities. Those that both my method and functional homogeneity would say are great. So I'm hoping that my method doesn't start saying that all these terribly, important communities are rubbish now, because that would be bad. I'm also going on to the things with CommWalker, so just accepted by CommWalker, and I'm going to have to look at those, because do they actually make sense? And the things just accepted by functional homogeneity, and then of course there's the things rejected by both. So we both think they're rubbish, so is that true? So first question is, am I overcoming what I actually said I wanted to overcome, which is inspection bias? You can't see the colours on here, you might be able to see them. So this is actually the hint P network, and it's laid out not really in a network way at all, but the nodes are laid out so that there's increasing similarity of the local environment. So I'm back like that picture I had at the beginning with the purple and green. So the things at the right-hand side are actually not very like their neighbours in terms of there's not much information about them in functional terms, and the things on the left-hand side, there's loads of information about them in functional terms. And then I've coloured the nodes if proteins are considered significant by both the original method, functional homogeneity, and by CommWalker. And you see this massive bias towards the left-hand end. So basically, we're picking up things which have lots of annotation on them, and we're saying that they're right. That's good. What's interesting is that I look at the next row down. This is the things that are considered significant only by CommWalker. I've now got this nice spread across the whole thing. So it is doing what I want it to do. I will then come on to whether that's useful, because of course, maybe these are all rubbish at this end. I should be aware that this could easily be rubbish and I could be generating garbage. You can also see, if you consider proteins considered significant only by functional homogeneity, that they have a very big tendency to just be at the left-hand side. So I am ruling out with my method some things which functional homogeneity would say, yes, yes, they're definitely significant, and we're saying, actually not compared to their neighbourhood. This is just to show it on a couple more data sets. So this is hint P on the left again, and this is the biogrid data set on the right. The reason the colours look denser on the right is just because there were so many more nodes, so if you put them in the same area, colour looks denser. And hint P has the Panday measure and the constant POTS model for the modularity optimization, and this is biogrid with the same things. And you can see exactly the same pattern happening again. So if I look at the third row down, things that are only accepted by CommWalker, I'm getting this spread across. And if I'm looking at things which are accepted by both, tendency to the left, things only accepted by functional homogeneity, tendency to the left. So this is what I wanted to see. I should say it looks exactly the same if I look at overlapping communities. It's very hard to make a pretty picture of that, so I haven't bothered. Because, it's just very difficult to think of a nice way of showing that. So it's doing what I want it to do. It identifies modules in less well-studied regions of the network. But the question that remains is, are those modules relevant? So mathematically, what I've done sounds sort of okay. It's not that exciting, but it kind of works. But I've got greater sensitivity in these poorly studied regions. But is it warranted? Could I have just made this up? As in, I've now managed to say that that's significant just because there's not much annotation there, but actually it's still just garbage and I probably shouldn't have done it. And it might be that I'm also causing myself a problem, because generally speaking, when you go to the less well-studied parts of any data, it's where all the errors are. So maybe I've just kind of invented some nice communities. So I'm going to go and try and validate this. And the data I'm going to use to validate it is gene expression data. So this isn't be used at all on the way through. It's another set of measures that people make on proteins. The good thing about this is they collect it genome-wide. So I have data for every single one of my nodes. The bad thing about it is that people think it's quite noisy and there are lots of discussions about exactly how you should use it. So it's not a perfect measure, But you would expect that if things are in the same functional grouping, they would co-express. What that means is they'd be around at the same time. So basically, I say they're being produced at roughly the same amount at the same time, or not being produced at the same time. That's what the co-expression means. So we've got a whole load of gene co-expression data. It's got 8,500 tissue-specific samples, and we evaluated them in very, very standard ways. Okay? And this is what happens. So if I look at the set which was both accepted, I get that red grouping. If I look at the set that was CommWalker accepted, I get the blue grouping. And if I look at the set that's only functional homogeneity accepted, I get the grey, and both rejected is the blue. So from this, it's clear that if they both say it's true, then actually that's got the best correlation of all. And that's nice, because these are two methods. They are related in a lot of ways, but they're not giving you identical results, and so that's good. What is more pleasing to us is that we have a whole set of communities, these kind of 5,000 odd, which are not well labelled functionally. So that's not 5,000 communities, I should be accurate here. That's 5,000 proteins within that set, which are not well labelled, so we don't know, which actually probably are coming from relevant biological communities. So they are worth testing in a sense of going and looking what the function is. This is probably a community or a module that has a novel biological function, and we don't know what it is, because actually it's not well labelled enough for me to tell you what it is. Whereas almost everything in that red box, I could have told you what it was before I began, before I did anything we've done here. What I also find quite interesting is the things that we reject but functional homogeneity accepts, actually there aren't that many of them, but also they are starting to be quite a lot worse. And so potentially we're right. Functional homogeneity is over-labelling, being overconfident in the areas of high annotation. It's saying, I've got lots of annotation here, so I'm getting reasonable functional scores, so I will just say, yes, it's a community, when actually it isn't. It's just because you've got lots of annotation, and so it looks a bit better than the average. And then it's very pleasing that the both rejected set actually is the lowest number of all, and so it all kind of makes sense. So I can't prove that this definitively works. I decided as this wasn't a biological audience, I wouldn't show you. I have 3 more slides which are examples that we've tested which are related to macrophage biology, which show that CommWalker works. In other words, we picked up novel modules related to that. But when I speak to a biological audience, that's considered to be definitive proof. I always see it as a kind of, here's a couple of case studies, which make me feel better about what I've done. So I kind of separate those out a little bit. So just to kind of finish off the bit about CommWalker, I think the nice thing about it is what you're really doing is getting increased sensitivity in poorly annotated areas without having to know any of the issues about the annotation that are going on. I don't have to know what the problems are with the annotation, I just have to know there is one. Yeah? So that is really helpful. And it means that I can actually attempt to pick up information in areas where there isn't very much annotation and be able to say, no, there is something interesting here. I don't know what that interesting is, but something interesting is there. I should state that, of course, if there was absolutely no information within a community, of course, I'd still fail because it would have a functional homogeneity score of 0, and probably so would all its random walks around it, because if there was nothing there at all. So there has to be some amount of information. That information can be very poor indeed. The other thing that I think is an issue within CommWalker, but is okay if we stay with relatively small communities, is, of course, it's probably gaining its kind of power by exploring the local neighbourhood. But at the moment, my random walks are not constrained in any way. So obviously, if I had a random walk of length 35 in a protein interaction network, I can go a really long way and be a long way away from that local neighbourhood. So something that we need to think about to see if it's necessary, and we haven't done yet, is whether we should constrain the random walks, because it probably would make things a lot better. We just haven't done it. But in my head, that's something that probably needs to be done. And the thing that I think is nice is that I think there are lots of systems, and there are certainly several others we've now come across, where actually this is a really good way of just evaluating your groupings, evaluating your communities, because it doesn't matter. We don't care how you broke your network up. We don't actually even care if you had a network underlying it, though obviously that's a lot easier for the whole random walk part. But we also don't care what your similarity measure is. Those are your decisions and the network type. None of this matters for this. So it could be very useful and applicable in almost any kind of setting where you have this problem, where you don't have full information on all the nodes, you have different depths of annotation, and you want to be able to identify what's interesting within it. So I'm now going to be cheeky, because I'm now going to talk about something we've been doing more recently, because we finished this a while ago.
Can I ask something?
Yes, of course.
So just going back to the measure that you actually proposed, which is rather a simple measure. It's just counting the number of times that your random walk improved or equalized, I guess, the score that you have inside, right? Is that number dependent on the underlying kind of topology of the network? So basically, depending on the structure of your module or the structure of the things that you have, like a full.
Yeah.
It is to, I mean, I think yes and no is my answer to that. So it is dependent on it because the relationship between the labels, I mean, there's a real relationship between nodes that interact with one another. So, and if you have a clique, then there's a relationship between all of them, which makes their labeling more likely to be like each other. But if you ask me the question as to whether the actual value of it is in some way related outside of the dependence of the labeling, then no, because it doesn't matter.
Because I was thinking, if the module is very isolated, you kind of expect basically to stay typically inside your module, and you'll get a distribution of values that is very much dependent on.
So we deliberately want to get a distribution that's dependent upon where you are within the network. That's kind of the point. I suppose if you have a structure of a network where you have a structure which breaks down very neatly into communities, so there's like a community here and then like one link back, and you delineate that as a community. If you use this as your measure, then obviously you would never find that to be significant, or it's very unlikely because nearly all your walks would stay within here.
If you plant a clique in a heritage rini, the distribution of your T depends on the P of the heritage rini.
Yes,
Yeah.
but I guess I'm seeing this as what we have here are relatively. So you saw the densities of networks. They're not very dense networks. Most of these networks we deal with, and we do not know what the underlying kind of production model of these is at all. I mean, I've worked on that. Loads of other people have done even better work on it, and we don't know. So I don't have a kind of. There's no graph model underpinning this at all.
No, but I was thinking if you could avoid just kind of it visiting itself, because you don't really want to visit itself, basically. The little peak you get, basically, which is self-visits, let's say. Well, you know where you are, so that's the kind of thing you want to smooth out, basically.
Yeah, I mean, I think the issue there is you want to smooth out visiting all of yourself, but if you have a community that's 20 nodes, I'm very happy for it to visit 10 of those and 10 other ones, because that would be a good comparator to say, is this 20 set better than the 20 set that would be half in here and half outside of here? So I think, yes, that's the kind of thing you could think about, which would be quite a good improvement to think about ways of saying, but then you'd have to, which I haven't thought about at all, would be how much of it would I let myself visit in some way? Because you're right, I mean.
That's interesting. Yep. Okay. Sorry.
That's all right. So I'm going to talk a little bit about a measure which we call net EMD. So there are lots and lots of ways out there of comparing networks. There's summary statistics for comparing them, so people would just say, does it have the same number of nodes? Is quite often used within the set of summary statistics. And there's really sophisticated, computation costly alignment-based approaches where you try and identify if these two nodes are identical or should be mapped to each other from two different networks, but that's computationally very costly. What I want to do here is cluster correctly networks that are different sizes and densities, but hypothesized to be structurally similar. So in model terms, that's a really simple thing. You'd say, I want to put all the Erdos Rennies over here, I want to put all the sticky models over here, I want to do that. And there are lots of other methods that think about this problem. And one of the reasons we want to think about this problem is actually coming back to what I just said about protein interaction networks. I'd love to know which group of networks they belonged to. Yeah? So what I'd like is a measure that was able to put real-world networks into the correct cluster they belong to, say with graph models, for example. And we wanted to try and develop something that was very simple and straightforward that would allow us to do this kind of thing. And what we're going to use is the Earth Mover distance, which I quite like because it's just a nice name for something. You build on the idea that a lot of these kinds of methods are built on, which is that the form or shape of a network property will be reflective of the topological organisation. So for example, that the degree distribution, and this is one that people have used for a long time, they say if the degree distribution is the same, then they're coming from the same thing. Yeah. Now that might not always be true, but it's one of the ways that you can think about it. And so if we kind of abstract this a little bit, we think of the underlying shape of a distribution as a property that's invariant under linear deformations, so translations and rescalings of the axis. That's useful to us here. So, if it's a bell-shaped curve, it'll still be one even if I sort of move it and move other parts of it around. And so the Earth mover distance is basically you can think of it, and it's kind of a nice way to think about it, which is, I guess, why it's called Earth mover, is the mass times the distance needed to transport one distribution onto the other. That's actually what you're calculating. And we're going to use this kind of EMD star where we're going to try and put the two distributions on top of one another to start with and scale them so they're the same. So you can think of a measure for two networks, G and G prime, and a given network feature T, which can have this Earth mover distance between them. And the nice thing about this is that it's symmetric and satisfies triangle inequality. So when I use the word distance, I mean distance, which very often is not true when we talk about measures of this type. And it's quite useful here. So this is just a kind of pictorial representation of what's going on. So those are, I have to get them right, Barabasi-Albert graphs on that side. These are Erdos-Renyi graphs. And then these are two which are the same size but from different types. So if I do what I've just described, which is look at their distribution, this is their degree distribution, I think. Yep. And I can rescale them. You can see that if I take two different, this is a fairly obvious result, two different sized networks, but I allow myself to rescale them to the same position of a particular graph type, their distributions look really like each other. So if you look at the Erdos-Renyi in the middle, they look very like each other, but one is 10 times the size of the other. Whereas even if I take two which are of identical size, I can't make them look like one another by doing this procedure. So these are different and these are the same. That's the property I would like. Now obviously, using something as simple as that property is not really going to cluster networks all the time. It will cluster simple things like this, but it's going to get me into trouble quite quickly. So what we want to be able to do is we're just going to have a sum of these, a sum of different network features. So for a set of features, we can define the measure corresponding like this. What we're actually going to use, that should be a 5 on the end there, not a 3. So graphlets up to size 5, sorry about that. This is only graphlets up to size 4 here because it takes too much space. But the automorphism of orbits of graphlets. So what that means is basically, I'm hoping most people here have seen this before, but just you're describing a graphlet as a small subshape within the graph, and then you're describing the properties of the different types of nodes you find within that subshape. And then you can have effectively something that looks like a degree distribution for each of those, so how often they occur at different degrees. So I can have, actually in this case, I could have 73 distributions. I will show you later that you don't need anywhere near that many, but we started out where everyone started out because it's about what you can calculate very easily, so graphlets up to size 5 and these different orbits.
Counting longer subgraphs takes a while, doesn't
It does, which is why we were quite grateful when we discovered we didn't need to, but we did start there. So I'm going to do 3 different synthetic network tests, and then I'm going to talk about some real-world data as well. So the 3 different synthetic network tests, there's the very easy one, just to make sure we're not doing something totally stupid, and also so we can compare to some of the other methods that use these graphlets. So I'm going to compare to other methods that use graphlets as their way of comparing. So the first one is actually just lots of identical networks. So can I find these if I do this? If I can't do that, then nothing else is going to work after that. Then I'm going to look at 2 sets, which RG2 and RG3, where I'm going to vary the size and the average degree. And if we get to RG3, the number of nodes and average degrees in networks both vary by 1 order of magnitude. So I still want to be able to cluster by model type, but the model could be 1 order of magnitude bigger in both degree and in actual number of nodes, or smaller. And then a data set that we've just constructed which has 10 different classes of real-world networks, so those are just 10 instances. And then there's a very large data set that was collected by JP Ornella which he published in 2012, of lots of real-world networks and synthetic networks. They're all different sizes. They go down to very small things and up to very, very large things, to see what would happen. And they're labelled by either how they were generated or the type of data they are. So they go from, there's authorship networks, protein interaction networks, circuit boards, all sorts of different things in there. So just to show you it in action as a distance. So this is net EMD on the left-hand side. And on the right-hand side is another measure, which is using the same 73 automorphous orbits, which is something called GCD. And then this is the kind of distance map we get. So looking at this, what I want is to see blue on the diagonal, because that's short distances between the things that are the same, and I'd like to see red off the diagonal, long distances between things that are different. So it looks like, on average, we're doing what we want to do, and we're actually getting much less sort of splurging between the types than we have here. So this is roughly what I want to see. But an interesting question here is actually how to quantify this nicely to be able to make a nice quantification. Because pretty pictures like this are always semi-convincing, but you don't actually know if the one on the left is better than the one on the right. It kind of depends how I pick my colour bar as well. So to measure performance, we're going to use something which other people have used, but others prefer some other measures. So we think this one is slightly better, but it doesn't matter that much. So for a given network, we've got kind of disjoint classes. So each of the networks belongs to a class. So it could be, if I'm doing synthetic networks, it's an Erdos-Renyi graph, it's a sticky model, it's a 3D graph. It doesn't really matter. And we'd like a network comparison measure to position in the same class closer to each other than networks in other classes. But we want to look at all of that at once, because we don't actually care what its exact neighbour is. We just know that all 10 of these belong together and not with the other 90. But I also, at the same time, don't want the other 90 to be modelled up wrong. I want to check that every 10 is correctly put together. So you can measure this in terms of the empirical probability that, if you like, the distance between your graph and any of the ones it should be next to is less than the distance between it and any of the ones it shouldn't be next to. And you can calculate the measure just like that. It actually looks, you can show that it's like the measure for a ROC curve, so a receiver operator characteristic curve. So if you get a p-value, if you get a p-bar of 1, that's perfect separation between the classes, and 0.5 is random. So what does that look like if I look at actual data? Well, that's net EMD on the left-hand side, and then these are other measures that use the graphlets for the comparison. So what I find slightly surprising, and I should just admit this, is that they all do very well on that first set of networks where I told you they're exactly the same size, with exactly the same density from exactly the same model, but they're not perfect at it. Even we're not. So we've done multiple tests on that one, because you can, but it's interesting to note that actually, even at that level of test where you'd think, surely you can get that right, that the standard methods don't. And I mean, we are within the error of getting 1, but we don't get 1 every time. Then if you make the problem a bit harder, and you try and separate out the network, so the pictures I just showed you on the previous slide were the equivalent for the RG2 case, which is quite difficult, and you can start to see that NetEMD is doing a bit better than the other methods, but they're all separating them pretty well. And even if you make the synthetic networks really, quite difficult to tell this would come from the same model, actually, all of the methods do relatively well at separating them apart. It's quite interesting. I mean, now NetEMD is showing that it's showing performance better than the others. When you start looking at real-world networks, you start to see other factors entering the behaviour. So you can definitively see that some of the methods cannot separate, or find it much harder to separate out these real-world networks. That however they're operating with their counts on the graphlets, they're failing to make this separation happen.
Can you tell me again, how do you know in real-world networks that they are the same?
So you've just by what they're labelled as. So that might not be a good definition. Okay. I should be careful here. So, for example, protein interaction networks could be one category, co-authorship networks could be another category, right. Transport networks could be another one.
Yep.
So the argument here is I would expect them to be closer to the things of their own class, but it might also be true that there would be some class mixing and you don't know what their nearest neighbour should be. And one of the reasons that we like NetEMD is because if it's a real distance, then the next closest thing really does mean next closest, rather than just random scoring that means that these ones are good and that everything else is bad.
The 6% that are off have you looked at them? Is there any characteristic which makes it not work that you see?
No. We can't work out why. I mean, we did set them all up so that effectively they have very similar average degrees, all the networks in that. So it's not totally trivial, because of course if I make the ERDAS already have a very different average degree from, then, so they've all got the same property there, but that still feels like you should just be able to do that. And it's interesting, I mean, I claim that we just about can, but it's interesting that these methods can't, I think, because it seems odd, because you think that must be the closest thing in there. What was sort of pleasing to us about this was that, well, sort of disappointing after being complicated and using all of the orbits, it turns out you really don't need to, or at least depends how hard your problem is as to how important that is. So these are just variants of the measure. There's some more real world networks at the bottom. So the bottom one there is separating out world trade networks into their time order. So can you get them in the right order? So this is the World Trade Network from each year. That's actually quite a hard problem, because it's not clear, you can have big jumps between certain years and almost no change between others. So we looked at different variants. So you could look at just graphlets just up to size 3 instead of up to 5, which we were doing before. And it turns out, if you look at the numbers there, we actually don't do much worse, which is quite scary, because that sounds like that's nearly all the information you need is in graphlets of up to size 3, so a triangle and a straight line is pretty much what I'm talking about there. And it's a bit better if I go up to graphlets of size 4. Some of the methods we were talking about on the previous slides took kind of samples of ego networks out of the network and looked at the orbits within those ego networks and did some of those. Turns out that really doesn't help us very much, and it's just a massive complication that we probably shouldn't have tried. And just to really hammer home that for some of the simple problems, just the degree distribution allows you to do an awful lot. So actually, just the degree distribution of the networks, I mean, it's fairly obvious, but it's interesting that we've come up with very complicated methods to separate out graph models. You don't need to, just rescale your degree distributions correctly, and then it will tell you which one's which. I don't think that should be a surprise, but it's interesting people put a lot of effort into showing pictures of how they can separate those out, when actually, and me included, we probably didn't need to. We could have been a lot simpler. So, but it does look like, as the problem becomes more complex, so the World Trade Networks are probably the most complex problem on here to actually try and do, putting more information into the measure is helping you. So using more graphlets becomes more important as you're trying to solve things which maybe have a finer resolution or are more complex to solve. But I wouldn't know that for definite. I wouldn't hold up my hand and say that's proof. I'd just say it looks like that. So the idea here was to have a very general network comparison methodology. And it's good because it successfully identifies similar networks at multiple scales. It's really nice and quick because I'm just comparing distributions. That's very, very fast indeed. It works even if I only use graphlets up to size 3, so 2 paths and triangles, so that's quite easy. And it has performance that's at least comparable to all of the state-of-the-art measures that you're using this kind of thing. But given that you can do it with such small things, actually you can compare things which have billions of nodes. That's totally possible within this architecture. And of course, I've compared it to, I suspect, my favourite graph feature, because I've done lots of work on graphlets before, so that's obviously where we started. But there's no limit on that. There isn't some, specialist cleverness that says graphlets are the right way to compare networks, or even necessarily the best way to compare networks. Depends on what you want to answer. So it might be graphlets are good for comparing biological networks, but they're rubbish for comparing for some other type of property you want to look for. And obviously you can apply this to whatever graph feature you like, as long as it has a distribution, because you can do exactly the same thing. And we've played around with that a bit, but we haven't actually sort of developed that much further along the way. So that is where I'm going to finish. I should politely say thank you to my research group. The guy standing at the very back there, looking slightly bald with blonde hair, is Malta, who did a lot of the work that I've just described here. And Lewis is standing right at the front with the red trousers on. Robert and Antol are both not in the photo, which is very rude of me. Gesina is somebody I did quite a lot of the work I've just described with, and some of it with Feng Zhu from USC, and some of the data, particularly for the first part of the work, came from UCB. So thank you very much.
Thank you. Maybe I have some more questions. I mean, an obvious question, given that Sophia's here, is could you use the kind of a graphon or some kind of graphon estimator to compare networks? Given that your degree distribution did so well, you would almost think if you were just to kind of estimate an underlying graphon and then compare the two graphons with each other.
The problem with the graphons is you still have to match the graphon estimates.
Yes.
So the blocks would have to be matched in blocks. I mean, one of the beauties here is that you've got rid of all of the matching problems and you just have the distribution.
Yeah.
Beautiful.
It's kind of like, just throw everything away, or as much away as you dare.
But you can obviously just order them via maybe degree distribution, degree of some, and so you could some.
Well, but that's where things, I mean, it's like you said that when the degree distribution is informative, it's gorgeous, right? But the whole reason you need the graph and all of that is that you think that actually a lot of the information is not in the degree distribution. So I can adversarially make up lots of examples where you have block models or incomplete beta distributions that you put together, so you get a flat marginal.
Yep.
So they all look like Erdős Rényi from the marginal, but actually there's a lot of hidden structure inside and do nasty stuff. So you should be.
And the
I mean,
graphlets would look different as well?
Sorry?
The graphlets would be informative in that case?
Yes,
You
yes,
can have
in
different
fact,
triangle stuff.
Yeah.
So
You'll
that's
have
be sufficient
flatlets,
to,
yeah.
okay.
If you look at the paper by Levina, Bickel, and somebody, they even do method of moments matching for the height of the block model approximation from wheels.
So I mean, I think I wouldn't say that you could just randomly pick any feature and it would separate it out beautifully. What I kind of like about it is you can pick any feature. And so if you get no separation, if the distances are all the same, then, and you think that there are differences between these networks, then obviously the feature you picked is not relevant. I mean, it's fairly clear cut. But it is, I mean, certainly for some of the things we're using there, you can come up with types of network where it would fail catastrophically.
I suspect you can no matter what, because I don't believe there's a UMP test for graph.
I mean, that's very much my feeling on this, is that what I wanted was something that was globally possible to run and also kind of plausible that you could choose different features. So, because also I have this feeling that we, like you said about the real world networks, what class do they belong to? Well, I'm trying to separate them by a set of man-made classes because people think that these ones are different to these ones are different to these ones. That might not mathematically actually be true.
Or even be the only clustering of those
Exactly.
collection of graphs. There might be another set that's also true.
Yes.
That's real, but
and contains different information, was useful to solve a different problem. So I think with both of these, but particularly with the second one, we were thinking very much along those lines about how do you develop something such that you would be able to think in that way. You know, obviously I've got to use a particular way of doing it first off, and as I said, I've done work on graphlets before, so graphlets it was.
right.
But I don't necessarily declare them to be the correct choice. at all.
We work on spectral embedding, so we did it. So we embed both graphs, and then we do the kernel test.
Yeah.
And that gives you a test statistic, and if that's big, then they should be in different groups. And it's not the same thing, and I suspect it's easy to see that the graphlets will have more power. You know, the spectral embedding kind of assumes some sort of exchangeability, so it's very good sort of across all alternatives as long as you're in the exchangeable setting. And I suspect that it's easy to construct a situation where the only difference is triangles versus not triangles, where you're gonna win hands down.
Yeah, and I think that's just part of the, it depends what you're trying to separate then, in my head.
There must be something good about using your distance measure, is that right? Because the other ones also use graphlets, the same
Yes.
graphlets.
Yes, and I mean they combine them very differently, so that most of the others don't combine them, they don't combine them in a kind of, any kind of, I'm trying to think of the right wording. They haven't thought about what the kind of value of the measure would be. It's like there are more or less of these graphlets, so we'll add one to this, it's bad or it's good.
Yeah.
Rather than saying, I'll start from the principle that what I would like is a distance,
Right.
or I'd like a metric. And the thing that interests me about this, and I have to say we haven't really explored this, you know, this is work that we're just kind of finishing off now, is because it's a real distance, there should be meaning to the thing you're second closest to, kind of thing. And so if you run this, yeah, and you put a protein interaction network in there, for example, against all of the possible graph models, previously people would go, well, the protein interaction network is closest under my scoring to this model, therefore it's this model. Yeah. Now I've got a distance that says, and I can start saying, well, these things definitely belong in the same model because their distance is always under some number because it's real in that sense. So if the protein interaction network is also under that number, it means something, as opposed to in their test environment where actually the number itself is potentially meaningless. So that's kind of where my brain is trying to think about it in those kind of ways to see, you know, does the second closest mean something, or can I actually show that it belongs within a particular grouping? And we haven't done any of that, and we kind of thought about those.
But you could use, if you do a nearest neighbor test.
Yes.
And think all kinds of things. So I have a question. So if you weren't interested in real problems for all your grants, but you were just interested in
I have to pretend, you know,
yeah,
it's very important.
but just in a property. So you used, let's say, you're then just using graplets up to size 3, so a path and a triangle.
Yeah.
You use the 2 marginals, but there's an earth mover distance on the joint distribution. I wonder if you could write down a case where the joint wins over the product of the marginals. I'm sure you could.
I'm sure you could.
And you're using the product of the marginals just because it's easier.
Yes, and it is literally because of that, because when we first started, we tried to look at the joint.
Okay.
And
And the extreme of graph theory tells you the support of the joint, because you can't have completely arbitrary.
Right, it's not going to be completely arbitrary, but there's a non-trivial joint.
and
Yeah, okay, so
we did
and by this you just mean computationally and numerically. Yeah.
just trying to work it.
So
And
the math says just do a joint.
yeah. And I think it's something we are going to come back to, because I think that, like you say, there are going to be obvious cases where that would be a much better way. And I have to admit, in my kind of head, I prefer thinking about the joint. I actually, but we've not done it. But yes.
Mm-hmm.
Will you get a lot of improvement, you think? Because obviously some of it doing already very well.
I think you probably won't for the kind of cases we're looking at there.
Probably not in real problems, but you can
Yeah.
easily construct a graph distribution such that.
Okay.
So I think that's a very accurate answer to that question. I suspect there are some real problems where it would be useful, but most of what we want to do in real problems is probably okay with what we've got.
Are you even thinking publication N+1, Karen?
It's just why else would I have come to this cellar? Yeah, that's right. And you could, the problem, of course, is back to your inspection bias. I mean, yes, there's a real problem, but you could search the world over for the real one and then publish that. Here's where it works, and not say that I did check 500,000 real
Yes.
problems. This is the first one I found, and that's the problem with all of science, right?
Yeah.
That's how
you do science.
No,
That's
exactly,
how,
just you.
that's how it would be done. That's,
Well.
yeah,
Do you have more questions?
that's a very nice talk.
Yeah.
Thank you.
Thank you.
And we-
I like it.